Question 1. While attending an in-service training on basic microbiology, a new technician in the Sterile Processing Department, SPD, learns about the cellular structures of various microorganisms. The lead clinical educator emphasizes that the complete destruction of a eukaryotic cell requires deactivating its functional center, which governs all cellular activities and heredity. Which cellular structure is the educator describing? A. Cell membrane. B. Nucleus. C. Cytoplasm. D. Extracellular polymeric substance, EPS. Correct answer B. Knowledge point explanation. Core rationale. In microbiology, the nucleus is defined as the functional center of a eukaryotic cell that contains genetic material and dictates all activity and heredity. Understanding this structure helps technicians grasp how biological reproduction works, even though the bacteria targeted in sterilization are prokaryotic and lack a true nucleus. Trap analysis. Option A is incorrect because the cell membrane is the semipermeable outer boundary that regulates the flow of nutrients and waste, not the control center. Option C is incorrect because the cytoplasm is the jelly-like substance filling the cell, not the heredity center. Option D is incorrect because EPS is the sticky slime layer secreted to form a biofilm, completely unrelated to the cell's internal governing center. Concept extension. While eukaryotic cells have a nucleus, bacteria, prokaryotes, contain a single circular chromosome located in the nucleoid region, which must be destroyed by sterilization parameters such as 270 degrees Fahrenheit, 132 degrees Celsius steam. Question 2. An SPD technician is investigating a moisture contamination issue involving flexible endoscopes. The infection control team identifies a specific gram-negative organism that thrives in moist environments, specifically residual water left in the endoscope channels. Based on typical environmental sources like water and soil, which pathogen is most likely responsible? A. Pseudomonas aeruginosa. B. Staphylococcus aureus. C. Mycobacterium tuberculosis. D. Geobacillus styrothermophilus. Correct answer A. Knowledge point explanation. Core rationale. Pseudomonas aeruginosa is a notorious gram-negative bacterium that is frequently found in water and soil. In the clinical setting, it exploits moisture, making inadequate drying of endoscope channels a critical risk factor for healthcare-associated infections. Trap analysis. Option B is incorrect because Staphylococcus aureus is a gram-positive coccus often associated with skin flora, not specifically a waterborne pathogen. Option C is incorrect because Mycobacterium tuberculosis is a bacillus causing respiratory disease, transmitted via airborne droplets, not primarily water channels. Option D is incorrect because Geobacillus styrothermophilus is a highly heat-resistant gram-positive spore used in biological indicators, not a standard waterborne clinical pathogen. Concept extension. To prevent the proliferation of moisture-loving bacteria, standard guidelines dictate purging endoscope channels with forced air until bone dry. Question 3. Which of the following best defines the specific scientific field that a sterile processing professional must master to effectively control and destroy invisible pathogens in the healthcare environment? A. The analysis of macrobiological disease transmission pathways. B. The scientific study of the nature, life, and action of microorganisms. C. The specialized study of human anatomy and operative surgical procedures. D. The chemical formulation of high-level disinfectants and sterilants. Correct answer B. Knowledge point explanation. Core rationale. Microbiology is explicitly defined as the scientific study of the nature, life, and action of microorganisms. This foundational knowledge allows technicians to understand the behavior, resistance, and vulnerabilities of pathogens to apply correct sterilization parameters. Trap analysis. Option A is incorrect because the field focuses on microscopic organisms, not macrobiological entities. Option C is incorrect because while anatomy is useful, it is not the study of the microorganisms themselves that we must destroy. Option D is incorrect because chemistry deals with the formulations, whereas microbiology deals with the living organisms those chemicals target. Concept extension. The term microorganism encompasses living things far too small to be seen with the naked eye, typically measuring around 1 to 5 microns in size. Question 4. When preparing a decontamination sink for instruments used on a patient diagnosed with herpes simplex, the technician must remember that this specific disease is classified as being caused by which type of pathogen? A. A highly resistant bacterial endospore. B. An abnormal transmissible protein. C. A viral infectious agent. D. A gram-negative bacillus. Correct answer C. Knowledge point explanation. Core rationale. Herpes simplex is scientifically classified as a viral disease, meaning it is caused by a virus, an obligate intracellular parasite that requires a living host cell to reproduce. Trap analysis. Option A is incorrect because herpes is not a bacteria, let alone a spore-forming one like Clostridium difficile. Option B is incorrect because abnormal transmissible proteins refer to prion, which cause diseases like Creutzfeldt-Jakob disease, CJD. Option D is incorrect because herpes simplex is not a bacterium. Bacilli are rod-shaped bacteria. Concept extension. Viruses are generally enveloped or non-enveloped. Enveloped viruses are typically easier to destroy using standard low-level disinfectants compared to non-enveloped viruses or bacterial spores. Question 5. During tray assembly, a technician spots bioburden deep within the box lock of a surgical clamp. If this instrument is packaged and stored on a wire rack at 68 degrees Fahrenheit with standard atmospheric exposure, pathogens that strictly require oxygen to metabolize and grow could thrive. How are these oxygen-dependent bacteria classified? A. Facultative bacteria. B. Aerobic bacteria. C. Anaerobic bacteria. D. Spore-forming mesophiles. Correct answer B. Knowledge point explanation. Core rationale. Bacteria that strictly require the presence of free oxygen to metabolize, grow, and reproduce are classified as aerobic bacteria. Exposure to standard room air supports their survival if moisture and nutrients are present. Trap analysis. Option A is incorrect because facultative bacteria can adapt and survive either with or without oxygen, whereas the scenario specifically describes organisms that strictly require it. Option C is incorrect because anaerobic bacteria thrive only in the absence of oxygen and would die or go dormant in standard atmospheric exposure. Option D is incorrect because while they might be mesophiles based on the 68 degrees Fahrenheit temperature, the primary classification for requiring oxygen is aerobic. Concept extension. The opposite extreme includes organisms like Clostridium tetani, causing tetanus, which are strictly anaerobic and can survive in deep, oxygen-deprived puncture wounds. Question 6. A technician is reviewing a clinical laboratory report for a contaminated orthopedic instrument set. The report identifies the bioburden as rod-shaped microorganisms that were found hiding within the serrations of the surgical clamps. Based on the 3 fundamental shape classifications of bacteria, how are these specific rod-shaped pathogens scientifically categorized? A. Cocci. B. Spirilla. C. Bacilli. D. Prion. Correct answer C. Knowledge point explanation. Core rationale. Bacteria are primarily divided into 3 main shape classifications: cocci, spherical; bacilli, rod-shaped; and spirilla, spiral. Bacilli look like tiny cylinders and are notorious for hiding in hard-to-clean areas of medical devices. Trap analysis. Option A is incorrect because cocci are spherical or round bacteria, not rod-shaped. Option B is incorrect because spirilla are spiral or corkscrew-shaped microorganisms. Option D is incorrect because prion are abnormal transmissible proteins, completely lacking cellular structure or standard bacterial shapes. Concept extension. Some species of bacilli are capable of forming highly resistant endospores, making rigorous mechanical friction essential during the decontamination phase. Question 7. During a routine steam sterilization cycle operating at 270 degrees Fahrenheit 132 degrees Celsius, the biological indicator fails, indicating microbial survival. The lead clinical educator explains that certain microorganisms are capable of forming a thick biological wall around themselves, enabling them to survive extreme heat, chemicals, and adverse conditions. What is the scientific term for these highly resistant structures? A. Endospores. B. Vegetative cells. C. Enveloped viruses. D. Cytoplasmic membranes. Correct answer A. Knowledge point explanation. Core rationale: endospores are highly resistant, dormant structures formed by specific bacteria when subjected to extreme environmental stress. This thick protective wall makes them the ultimate benchmark for validating sterilization efficacy. Trap analysis: option B is incorrect because vegetative cells are actively growing and reproducing bacteria, which are easily killed by low-level disinfection and standard heat. Option C is incorrect because enveloped viruses have a fragile lipid outer layer that makes them highly susceptible to environmental destruction, not highly resistant. Option D is incorrect because the cytoplasmic membrane is a delicate internal cellular structure, not a thick survival wall. Concept extension: Geobacillus stereothermophilus is the specific endospore used in biological indicators to test the lethality of steam sterilizers. Question 8. To correctly identify unknown pathogens recovered from soiled surgical instruments, clinical laboratories rely on specific staining techniques that classify bacteria by color changes in their cell walls. What are the two most common processes designed to achieve this classification? A. Silver stain and Giemsa stain. B. Gram stain and acid-fast stain. C. Methylene blue stain and crystal violet stain. D. Primary stain and counter stain. Correct answer B. Knowledge point explanation. Core rationale: the Gram stain and the acid-fast stain, such as the Zeil-Nielsen stain, are the two most fundamental diagnostic processes used to classify bacteria based on their distinct cell wall compositions and color retention properties. Trap analysis: option A is incorrect because while these are valid tissue stains in pathology, they are not the primary, most common methods for bacterial classification in standard microbiology. Option C is incorrect because these are individual chemical dyes used within staining processes, not the names of the comprehensive two-part classification processes themselves. Option D is incorrect because primary stain and counterstain are generic steps within a staining protocol, not the specific names of the classification tests. Concept extension: the Gram stain divides bacteria into purple gram-positive and pink/red gram-negative organisms, which heavily dictates the selection of appropriate antimicrobial agents and disinfectants. Question 9. A technician is processing a trauma set used on a patient with a severe, deep tissue puncture wound. The surgeon suspects contamination with the highly dangerous pathogens responsible for tetanus and botulism. Because these specific organisms thrive deep within wounds and do not require oxygen to grow, how are they scientifically classified? A. Aerobic bacteria. B. Facultative bacteria. C. Thermophilic bacteria. D. Anaerobic bacteria. Correct answer D. Knowledge point explanation. Core rationale: tetanus and botulism are caused by pathogens, like the Clostridium genus, that do not require free oxygen to survive and reproduce. Therefore, they are strictly classified as anaerobic bacteria. Trap analysis: option A is incorrect because aerobic bacteria strictly require the presence of oxygen to metabolize and survive. Option B is incorrect because facultative bacteria can adapt to survive with or without oxygen, but the question specifically targets organisms defined by their lack of need for oxygen. Option C is incorrect because thermophilic refers to a temperature preference, extremely hot, 122 degrees Fahrenheit to 158 degrees Fahrenheit, not an oxygen requirement. Concept extension. Thorough mechanical cleaning with detergent and water is critical to remove the protective bioburden that creates microscopic, oxygen-depleted environments where anaerobes could thrive on instruments. Question 10. When selecting the appropriate chemical disinfectants for the decontamination area, the Sterile Processing Department, SPD, staff must understand the cell wall structures of common pathogens. Using the standard gram stain classifications, which specific category do common healthcare-associated organisms like Staphylococcus, Enterococcus, and Streptococcus belong to? A. Gram-negative bacteria. B. Gram-positive bacteria. C. Pathogenic prion. D. Enveloped viruses. Correct answer B. Knowledge point explanation. Core rationale. According to standard microbiological classifications, Staphylococcus, Enterococcus, and Streptococcus possess thick cell walls that retain the primary purple dye, classifying them unequivocally as gram-positive bacteria. Trap analysis. Option A is incorrect because gram-negative bacteria, such as Pseudomonas or Escherichia coli, have a thinner cell wall with an outer membrane and stain pink or red. Option C is incorrect because prion are non-living, abnormal proteins that do not undergo gram staining at all. Option D is incorrect because viruses are subcellular organisms that lack a cell wall entirely and cannot be gram-stained. Concept extension. Although gram-positive bacteria have thick cell walls, they are generally more susceptible to standard low-level disinfectants than gram-negative bacteria due to the absence of the protective outer lipopolysaccharide, LPS, layer. Shutterstock.
探索
Question 11. While monitoring the rapid exponential growth of a bacterial culture in an incubator set to 98.6 degrees Fahrenheit 37 degrees Celsius, a technician observes a single parent cell dividing into two identical daughter cells. Because this process can occur every 20 minutes, soiled instruments must be transported to the decontamination area immediately. What is this specific bacterial reproductive process called? A. Repopulation. B. Binary fission. C. Meiosis. D. Bilateral reproduction. Correct answer B. Knowledge point explanation. Core rationale. Binary fission is the primary method of asexual reproduction for bacteria, where one cell physically splits into two equal, identical cells. Understanding this exponential growth underscores the urgency of prompt instrument cleaning to prevent massive bioburden buildup. Trap analysis. Option A is incorrect because repopulation is a generic term for a population growing again, not the specific biological cellular division process. Option C is incorrect because meiosis is a type of cell division for sexual reproduction in eukaryotes, not bacteria. Option D is incorrect because bilateral reproduction is a fabricated, non-standard term in microbiology. Concept extension. This rapid reproduction requires a nutrient-rich environment, moisture, and specific temperatures, which are often perfectly provided by blood-soaked surgical instruments. Question 12. During a complex neurosurgical procedure, a craniotomy set is exposed to central nervous system tissue from a patient suspected of having Creutzfeldt-Jakob disease, CJD. The Sterile Processing Department, SPD, must follow rigorous extended sterilization protocols because the infectious agent is highly resistant to standard processing. What type of infectious agent causes this disease? A. Prion. B. Anaerobic spores. C. Enveloped viruses. D. Gram-negative bacilli. Correct answer A. Knowledge point explanation. Core rationale. Creutzfeldt-Jakob disease, CJD, is a fatal neurodegenerative condition caused by prion, which are abnormal, misfolded, and highly infectious proteins. Because prion are not living organisms, they exhibit extreme resistance to conventional sterilization methods. Trap analysis. Option B is incorrect because while spores, like Clostridium, are highly resistant, CJD is not caused by a bacterium or its spore. Option C is incorrect because enveloped viruses are actually very easy to kill and do not cause CJD. Option D is incorrect because gram-negative bacilli are bacteria, whereas the CJD agent completely lacks genetic material and cellular structure. Concept extension. Prion have a strong affinity for binding tightly to metal surfaces, making specialized extended steam sterilization cycles, e.g., 273 degrees Fahrenheit per 134 degrees Celsius for 18 minutes, or specific chemical treatments necessary. Question 13. In the foundational terminology of sterile processing and infection control, what is the exact definition of the state of being soiled or made impure by contact with infectious organisms or other hazardous materials? A. Sterilization. B. Bioburden. C. Contamination. D. Disinfection. Correct answer C. Knowledge point explanation. Core rationale. Contamination is explicitly defined as the state of being soiled or infected by contact with infectious organisms or other materials. This concept dictates that any item dropped on the floor or exposed to unsterile conditions must be considered unsafe. Trap analysis. Option A is incorrect because sterilization is the complete destruction of all microbial life, the exact opposite of contamination. Option B is incorrect because bioburden refers to the actual number of microorganisms present on an object, not the general state of being soiled. Option D is incorrect because disinfection is the process of eliminating most or all pathogenic microorganisms, except bacterial spores. Concept extension. To prevent cross-contamination, healthcare facilities employ designated dirty and clean areas, strict one-way workflow, and positive/negative air pressure differentials. Question 14. When evaluating the pore size specifications of a new high-efficiency sterilization wrapper, the lead clinical educator explains that the material must act as a microscopic shield. To effectively prevent the penetration of common pathogens, the wrapper must be engineered to block microorganisms that typically fall into what size range? A. 0.1 to 0.5 microns. B. 1 to 5 microns. C. 10 to 50 microns. D. 50 to 100 microns. Correct answer B. Knowledge point explanation. Core rationale. Most standard pathogenic bacteria encountered in the healthcare environment measure approximately 1 to 5 microns in size. Sterilization packaging and high-efficiency particulate air, HEPA, filters are meticulously engineered around this specific biological measurement to ensure physical barrier integrity. Trap analysis. Option A is incorrect because this range is far too small for typical bacteria, aligning closer to viral particles which are subcellular. Option C is incorrect because organisms of this size are excessively large for standard single-celled bacteria. Option D is incorrect because 50 to 100 microns is approximately the width of a human hair, which is visible and massively larger than bacteria. Concept extension. A single micron, micrometer, is equal to approximately 1/25,000 of an inch, illustrating why even the smallest pinhole in a sterilization wrapper compromises the entire instrument set. Question 15. While certain bacterial endospores can remain dormant and survive harsh environmental extremes for years, pathogens in their active, multiplying phase are highly susceptible to destruction. During this active phase, direct exposure to environmental factors like sunlight is rapidly lethal to the cell. What is this active growth stage called? A. Incubation stage. B. Vegetative stage. C. Dormant stage. D. Sporulation stage. Correct answer B. Knowledge point explanation. Core rationale. The vegetative stage refers to the active, growing, and metabolizing state of a microorganism. In this state, pathogens lack thick protective outer walls, making them highly vulnerable to environmental stressors like ultraviolet, UV, light from sunlight, heat, and standard chemical disinfectants. Trap analysis. Option A is incorrect because the incubation period is the time between exposure to an infection and the appearance of the first symptoms, not a cellular life stage. Option C is incorrect because the dormant stage is the inactive survival state, like a spore, which is precisely the opposite of the active vegetative state. Option D is incorrect because sporulation is the transitional process of a vegetative cell forming a protective spore under stress, not the active growth phase itself. Concept extension. Low-level disinfectants are generally effective at killing vegetative bacteria on non-critical items, but they are ineffective against highly resistant lipid-lacking non-enveloped viruses and bacterial spores.